everyone, I'm Dr. Nasreen Peer, a researcher and lecturer at Stellenbosch University, and today I'll tell you more about our project, Merging Scientific Study and Indigenous Knowledge in the Inhambane Bay Mangroves in Mozambique. As you can see, it's quite a long list of collaborators, both from South Africa and Mozambique, and it is, thankfully, a list that keeps growing. Inhambane, for those of you who don't know, is a city in southern Mozambique, located on the eastern shore of the Inhambane Bay. The Inhambane Bay itself is a well-mixed mesotidal estuarine bay, and if you'd like to read more about the hydrology of the bay, you can find this paper by Solana et al., published 2020. So local communities live all around the bay, and here you have a bird's eye view. And these are mostly subsistence fishing and harvesting communities. What do they fish and harvest? Well, they catch fish both by boat and on foot. They harvest crustaceans and mollusks, and they harvest mangrove wood. They use this wood both for construction as well as for firewood. So other activities around the bay include tourism, pastoralism, and subsistence agriculture. So as you may have gathered, mangroves line the bay as well as seagrasses. You can read more about these in depth in papers by Amone Mabuto et al. 2017 and a paper by Barbosa et al. 2001. If we were to look closer at these two papers, you'll see that Barbosa et al. Recorded a 1.2% deforestation rate of mangroves in Inhambane Bay between 1972 and 1990. Amone Mabuto et al. Recorded an almost 50% decrease in seagrass cover in the bay as well between 1992 and 2004. However, local communities do understand the ecological importance of mangrove and seagrass habitats. They've identified these as nursery habitats for both reef and estuarine fish. And as such, they've developed several community no-take zones. You'll see these marked as stars on the map. There are stories of these no-take zones going back quite a few generations, in fact. They've also developed some mangrove replanting initiatives. And here's an example of a community nursery. All these saplings and seedlings will soon be replanted. So during discussions between us as scientists and communities, the question came about how effective are our efforts, our conservation and replanting efforts. And as scientists, we have the tools to help answer that question. So in line with this, we developed three main aims for our project. The first being to describe the mangrove habitat and ecology of the Inhambane Bay. Since the mangroves hadn't really been studied since 2001, and the mangrove fauna haven't been studied for decades, we thought it best to start here. The second aim was then to assess the efficiency of the no-take zones. The third was a bit more abstract and came about so that we could also learn some lessons from this project about how we go forward in our careers engaging with other communities in Southern Africa. And this is to explore the relationship between scientists and communities. Now, it was important for us to use both our scientific methods as well as to capture and use indigenous knowledge during this project. For our aim 1, which was to look at the mangrove ecology, we had a set of ecological methods and a set of social data collection methods. So for the ecological data collection, we started off with human use scoring, and here we really wanted to classify our sites. So were they more pristine, were they heavily used, were they urban? Then we collected the usual suite of parameters, being your physico-chemical parameters. We counted diversity and abundance of mangrove invertebrates using macrofauna quadrats, and we conducted tree surveys. Dr. Celia Makamo, who looks at soil carbon through Mozambique, collected a few soil carbon cores as part of this project. And then for the social data collection, and I'll be honest, this is not really my area of expertise, but we have a few great social science collaborators. Here we used field notes, semi-structured interviews. We held one community gathering, and hopefully soon we'll use a participatory mapping approach as well. So to classify the site in terms of human use, we looked at 5 characteristics. That is, was the site a no-take zone? What was the intensity of harvesting? What building materials were used to build houses? What kind of boat traffic did the area experience? Was it fueled traffic or was it a paddle boat? And lastly, how close were buildings to the mangroves? Using the scoring system, we classified our 6 sites. We found that we had 2 urban sites. Those were the mangroves in the cities of Inhambane and Mashish. And I apologize because this is actually not really labeled properly. Um, then we had 2 heavily used sites, that being Marambone, and Marambone being the blue dot in the north, and Ginziwe being the blue dot further south. And lastly, we had the 2 fairly pristine sites, being Giua in the far south and Morumbene in the far north, the purple dots. Of course, these pristine sites still do see human traffic and human use, but they are fairly pristine compared to the other sites. What I need to mention as well is that these results are currently being written up by Shaquille Benjamin, my honors student. So he used or developed the human use scoring system, and then together with that, he looked at the invertebrates that we'd recorded. He came up with a list of 25, and he wanted to see if there was a distinct grouping of invertebrates between zones. So he found that there actually was a distinct grouping. And then he went one step further and wanted to look at whether or not any of these could be suitable bioindicators for mangrove health, or maybe degrees of human use or human impact in mangroves in Mozambique. And he used the inval function to look at were any of these invertebrates potential bioindicators. So for zone 1, he found 2 fiddler crabs to be quite ideal, as well as 1 sesarmid. And for zone 3, he found the mangrove tree climbing crab to be a good indicator of urban sites, as well as the army crab Datilla fenestrata and the giant mangrove whelk Terebralia palustris. Now this was interesting for me because from my experience, Terebralia doesn't really do well in degraded sites. However, in Inhambane Bay, we record crazy high numbers of Terebralia and Datilla actually in our urban sites. So we wanted to see if the tree data sort of corresponded to these zones of human use. Zone 1 being the more pristine, Zone 2 being heavily used, and Zone 3 being urban. Before I get into that, you'll see that I've kind of just plotted the salinity very crudely above the tree data. And I just thought it's interesting, or I found this interesting, is that even though the estuary is quite well mixed and salinity is fairly stable throughout or fairly uniform throughout, there's one site, Marambone, where salinity goes up to probably beyond 70. The reason the graph only goes up to 70 is because that's the limit of the probe. We did hear that the site was used as a salt harvest site, which was quite cool. Coming back to the tree data, you'll see that the 2 species with the highest importance values were Avicennia marina, which we are all quite familiar with here in South Africa. It's a fairly common mangrove species. And then the second one is Soneratia alba, which we don't get in South Africa. It's also known as the apple mangrove, and I've just put in a couple of pictures on the side so you can see the fruit, as well as the very strange knobbly roots. So at our 2 urban sites, Mashish and Inhambane, you'll see that These sites were dominated by Avicennia marina and Soneratia alba, whereas as you move towards the more pristine sites, you see other species coming through, such as your Cereops tegal, Rhizophora macronata, and Bruguiera gymnorrhiza. So you can clearly see the patterns between urban, heavily used, and pristine. And what's interesting to note is that the pristine sites, Avicennia marina, still sort of has a high importance value. But that is because at these sites you get these massive, sometimes 15-meter tall Avicennia trees with huge trunks. You can see some pictures of us um, in some of hugging or sitting in some of these trees. So it's really quite remarkable to actually see these in person. And when it comes to community insights, we've collected loads of data from our field notes, a community gathering, as well as some semi-structured interviews. These are still in the process of being analyzed, but what we hope to gain from these data is firstly to understand if there have been changes in mangrove cover over time or changes in mangrove use over time. We also want to understand the community's perceptions of mangroves. Do they find them a nuisance? Do they find them useful? And so far we find that this seems to vary from urban to more pristine settings. And lastly, we want to have a brief insight into the community-based management of these mangroves. Moving on to aim 2. Here we wanted to look at the efficiency of the no-take zones. So we haven't started collecting these data yet, but we do have, again, our ecological data collection, which consists of environmental DNA assessment to look at diversity, and then BRAV analyses, which is baited remote underwater video, to look at more relative abundance. And because here we were looking specifically at fish no-take zones, we wanted to compare the fish communities between open zones and no-take zones. And it was important that the methods we used were non-invasive. So we didn't want to catch the fish. We didn't want to really disturb the area. So environmental DNA assessment allows us to do this, along with BRAV analyses, of course. And then our social data collection, which again would come from our field notes during field trips with our community guides, and then stories told during community gatherings, and questions and answers during those same gatherings, as well as semi-structured interviews and hopefully participatory mapping. So we started our BRAV trials in Feb 2020. Obviously, we could not continue due to the COVID lockdown, but we had varying success. So here you see Senor Nyamusua, who is one of our main community collaborators, and with our very homemade BRAV setup. And we've kind of designed this so that it's easy to disassemble, easy to take with us when we drive or fly up there, and easy to use, easy to put together, so that even once we leave or once there's no research being conducted, communities can still sort of use these methods. You can see the little GoPro attached at the end and some little Lethrinid juveniles coming up to the canister, which was quite empty, and a crab in the background. So some sites were wonderfully clear and we got great footage. And it's also great to see the seagrass habitat being used by juveniles. Other sites, because we had just visited just after the rain, we got nothing. But we have marked the good sites. We will continue to look for sites, and this is an ongoing process. And then for our eDNA sample design, we plan to sample 3 to 4 clusters. Within each cluster, you'll find a no-take zone and a fishing zone, and we'll collect samples from each of these. So I can try to give you a quick run through of the eDNA methodology for those of you who are unfamiliar. I will say again that this is not my area of expertise, but our collaborator, Sophie van der Hayden, is leading the sample design and methodology with this section. So environmental DNA sampling is based on the premise that as organisms move through a substance, they leave traces behind. So little particles of skin, tissue, mucus, etc. So you collect a sample of an environmental sample. In this case, we'll collect a water sample, and we sequence all the DNA within this water sample using a metabarcoding technique. And then you run all these sequences or blast these against a barcoding library, and you can create a list of species that have passed through your sample. Here we are partnering with the local NGO Ocean Revolution as well as Nature Metrics, which some of you may be familiar with. And we realized that we both kind of had similar aims. So Ocean Revolution is very focused on helping the community to conserve and to monitor their conservation efforts. Um, so Nature Metrics came on board with funding to help them, and we, of course, are working with the community directly. So we decided to partner up in order to sample more no-take zones and to help build a better reference library. And then again, with the community insight, what we're hoping to investigate is firstly the roles of the community fishers, especially looking at things like gender and age and how these have changed over time. And then again, the change in catch and effort over time. We hear a lot about how catches are really bad these days, or catches are bad in years where there's no rain, but it would be great to document this a bit more formally. And then we wanted community perceptions about community-based management. You'd think it was a popular thing, but not everyone feels as positively about it. And then I think one of the broader add-ons or aims is to look at climate change impacts and how to build resilience with communities. And this is an aim of Juliana Come, who is based within your department, supervised by Bernadette Snow, and co-supervised by myself. And then for the last aim, to look at the scientist-community relationship. This is not so much a formal thing as questions that we bear in mind and discuss often amongst ourselves, with collaborators, with communities. And these are questions such as, what do we actually give to communities in return as scientists? How do communities even feel about scientists? We found that communities actually have quite a bad perception of scientists. Obviously, this is not great because we don't want to cause conflict as scientists, but also because, cooperation and collaboration, it gives everyone more. And then, oh yes, so if communities don't feel great about scientists, often there's a breakdown in honest communication. Then what role do other stakeholders play? For example, government departments, NGOs, policing authorities. And lastly, how can we involve subsistence communities in science? So we go on a lot about citizen science and science communication, and these are popular terms, but practically what does that mean, especially in a Southern African context? So in November 2018, we held a community Bio Blitz in collaboration with Ocean Revolution. This was such a fantastic thing to be part of as a scientist. We had two schools come in. We gave the kids containers, and they went off into the mangroves to collect whatever they could find. We then helped them to identify the animals and plants that they brought back before, releasing everything back into the mangroves. What was really cool is that we were joined by several other community members, including a bunch of local fishermen. So they brought with a few cool things that they'd picked up on their fishing trips, and they told us stories of their fishing trips. They, spoke to us about the changes in their catch that they've seen over the years. And we had a couple of cool gadgets as well. So at the bottom you can see a homemade smartphone microscope. And here I'm working with some of the employees of the fisheries, the government fisheries department. I don't remember what we were looking at, but we were trying to get it in focus so that the kids could come have a look. So yeah, some of the weird things we saw, obviously some things were quite common, like your salt marsh, um, and a couple of snail species, but then the fishermen brought us things like seahorses and really strange crabs that they'd obviously netted from greater depths. It's important to note that these are things they'd collected already, so it's not like we had encouraged them to collect it just for this BioBlitz. And then what was really fantastic was the stories and input from community elders and fishers. One thing we found with our interviews is that most people actually have no recollection of things, or are not as free to share things from before the war ended, which we did not expect, or I did not expect. But during this Bio Blitz, the community elders were a lot more open and a lot more, they told their stories so easily, especially because they were talking to the kids from their communities. And lastly, we kind of made or mapped out our methods, and we wanted to see, okay, we generally focus on scientific objectives, but what about the community-focused objectives? How do our methods contribute to community-focused objectives? So if we just consider our three main aims, the direct objectives, you can see aim 1 is a science-focused objective where we want to study the mangrove ecology and the change over time. Still using both indigenous knowledge or social data and your more ecological data. And then your community-focused objectives would actually fall under aim 2, so the efficiency of community Notake zones, because here we're providing information that has been requested by the community that will be of use to them. And then indirectly, this kind of covers our aim 3. You can see that the scientists get a better understanding of successful community conservation dynamics, as well as community science dynamics. And the community-focused objectives is that there is a knowledge exchange between the indigenous knowledge holders and the scientists with the appropriate recognition. And the one thing that keeps coming through is how important this is for communities. They always talk about scientists coming in, interviewing them, doing projects, and kind of leaving, without giving anything back or without even so much as acknowledgement. So what they really indirectly want from our relationship is appropriate recognition, and not just knowledge exchange between scientists and communities, but also between community members. So often there isn't this conversation that happens between elders and children, but community gatherings or certain events allow more focused discussions around these topics. So here I feel like I need to note that until I, went to Mozambique, my training and my experience in ecology in South Africa was very much scientific focused, very much focused on the environment itself. Until I moved to Mozambique, I had no idea just how important it was to look at the social aspect. And obviously, the main lesson we learned, or the first lesson from an ecological perspective, is the huge variation in mangrove states, use, and management between South Africa and Mozambique. So in South Africa, we tend to conserve through, or focus on conservation through parks where you have a top-down control. But in Mozambique, more power is given and more support is given to communities so that there is a bottom-up control and management of environments and resources. So this was really interesting, especially because we are neighboring countries. And then when it came to the science community relationship, we learned that it was important to give proper acknowledgement of communities in research, something that we as South Africans very rarely do. So definitely a big lesson for me and for some of my collaborators going forward. And what was interesting here is the lesson that, successful conservation actually comes from building on what works for the community, not going into a place and saying, well, ecologically we've figured out that this is what we need to do, this is what you have to do, this is where you can take from, this is where you can't take from. It's first going in and listening and then building from that. Because if you go in with a prescriptive attitude, chances are it's not going to work. The last one is a bit again was a bit of a lesson towards my own attitude and behavior. So many of you might have seen these conservation experiences advertised, come to Mozambique for the summer and, contribute or volunteer and help locals conserve the whale shark or whatever it is. But Often these are led by foreign conservation NGOs, and foreign conservation NGOs in East Africa are extremely problematic for a number of reasons. Firstly, they are usually led by self-interest as opposed to a real vested interest in the area. What does this mean? So often you'll find that the volunteers and the people working for these organizations want to live in paradise, or they want to conserve the whale shark. These are all romantic ideas of conservation. You know, they're not going in with the idea that they want to help develop capacity or help empower. In fact, often their actions are disempowering rather than empowering. You also find that these NGOs disregard permission, be it governmental, be it community, um, and there is a large ignorance of power dynamics without even people trying to understand how they fit into a system in a foreign country. As scientists, we need to be extremely careful of not painting a similar picture. Um, and luckily for us, there are some easy ways to do this. And for me, the first and foremost is to always ask and listen first. So if you move into a new area or you're about to study a new area, you know, find out which communities rely on the area or move through the area, and you ask your questions first and you listen first. Then is to establish a collaboration or cooperation, make sure that everyone is equally acknowledged, be it through co-authorship. Um, and this involves not just scientists, but also the non-scientist stakeholders that use the area. Mapping out and discussing benefits has been really great for me, or really beneficial for me and our project. And then lastly, especially, this one is especially if you're working in a foreign country, is student co-supervision, mentorship, and student pairing. So for me, it was important not to just have South African students go over, study in Inhambane Bay and, return, but to help develop capacity of Mozambican students. So we've been trying to raise funds. All my South African students are co-supervised by Mozambican collaborators, and this helps to build your relationship and build a partnership really in many, many different ways. Okay, here I just have to acknowledge my collaborators, all our community collaborators from the Community Council of Fishers, my collaborators and students at Stellenbosch University and the University of Eduardo Mondlane and Nelson Mandela University. And then a huge thanks to our partners, Ocean Revolution and Nature Metrics, and to our primary source of funding, the National Research Foundation. Thank you for listening. I hope you enjoyed my talk.